Ovulation and oocyte transport. The process begins with the female ovarian cycle, driven by a surge in luteinizing hormone, or LH, the dominant Graafian follicle ruptures. This ovulation event expels the secondary oocyte, which is arrested in metaphase 2 of meiosis. The oocyte is released along with its surrounding protective layers, the inner glycoprotein matrix called the Zona Pellucida, and the outer cellular layer called the corona radiata. The finger-like fimbriae of the fallopian tube sweep over the ovary, creating a fluid current that draws the oocyte into the infundibulum. Cilia and peristaltic contractions move the oocyte to the ampulla. The oocyte remains viable in the ampulla for only 12 to 24 hours. Sperm deposition in transit. Simultaneously, hundreds of millions of spermatozoa are deposited into the vaginal canal. A mature spermatozoon consists of a head containing condensed haploid DNA, a midpiece packed with mitochondria for energy production, and a flagellum for motility. The vaginal environment is highly acidic, with a pH of roughly 4.0, which is lethal to sperm. Seminal fluid acts as an alkaline buffer to temporarily neutralize this acidity. The surviving sperm must navigate the cervix. Under the influence of high estrogen levels during ovulation, cervical mucus becomes thin and watery, forming microscopic channels that guide morphologically normal sperm forward while filtering out abnormal variants. Capacitation and uterine survival. Once inside the uterine cavity, sperm undergo a critical physiological activation called capacitation. As sperm swim through uterine fluids, inhibitory proteins and cholesterol are washed away from their plasma membranes. This destabilization increases membrane permeability to calcium ions, leading to hyperactivated motility. The flagellum beats with higher amplitude, giving the sperm the physical force required to penetrate the egg later. During this transit, the female immune system mounts a massive inflammatory response. Neutrophils and macrophages flood the uterus, destroying millions of sperm via phagocytosis. To reach the correct fallopian tube, the remaining sperm rely on chemoattraction and thermal gradients to navigate toward the ampulla. Out of the initial millions, only a few hundred sperm survive to reach the oocyte, the acrosome reaction, and penetration. When the surviving sperm reach the ampulla, they must breach the protective layers surrounding the oocyte. Sperm use hyperactivated motility to push through the outer cells of the corona radiata. Upon reaching the inner layer, specific proteins on the sperm cell membrane bind to ZP3 glycoprotein receptors on the zona pellucida. This binding triggers the acrosome reaction. The acrosomal vesicle at the tip of the sperm head releases proteolytic enzymes, specifically acrosin and hyaluronidase. These enzymes locally digest the zona pellucida, allowing the sperm to tunnel through and make contact with the oocyte's plasma membrane. Prevention of polyspermy and fusion. The moment the single champion sperm membrane fuses with the oocyte membrane, it triggers a critical event to prevent polyspermy, which is the fertilization of an egg by multiple sperm. This defense mechanism is called the cortical reaction. Membrane fusion triggers a massive intracellular release of calcium ions across the oocyte. This calcium wave causes cortical granules, located just beneath the oocyte membrane, to undergo exocytosis. They release enzymes into the peravitallane space that structurally alter the zona pellucida, destroying all remaining ZP3 receptors and hardening the outer shell. This completely blocks any other sperm from entering. The entry of the sperm also signals the oocyte to complete its second meiotic division, expelling a second polar body. Cleavage and blastocyst formation. Inside the cell, the male and female pronuclei fuse their genetic material during a process called amphimixis. This restores the diploid number of 46 chromosomes, officially forming a zygote. The zygote immediately enters the cleavage stage, undergoing rapid mitotic cell divisions without increasing in overall size. By day 3, these divisions form a solid ball of 16 identical cells, known as a morula, which remains contained inside the rigid zona pellucida. By day 4 or 5, fluid accumulates inside the cell mass, creating a central cavity. The cells now differentiate into two distinct populations, an outer layer called the trophoblast, which will form the placenta, and an inner cell mass, which is embryologically destined to become the embryo. The structure is now officially termed a blastocyst, implantation, and endocrine signaling. By day 6, the blastocyst enters the uterine cavity and undergoes hatching, shedding its restricting zona, pellucida. The exposed trophoblast cells attach to the endometrial lining of the uterine wall, typically along the posterior fundus. Upon attachment, the trophoblast differentiates into 2 layers, an inner cytotrophoblast and an outer invasive syncytiotrophoblast. The syncytiotrophoblast secretes lytic enzymes that digest the maternal endometrial tissue, allowing the blastocyst to burrow deep into the uterine wall and establish a connection with maternal blood vessels. This is the process of implantation. To maintain the pregnancy, the syncytiotrophoblast begins secreting human chorionic gonadotropin, or HCG. This hormone enters the maternal bloodstream and rescues the ovarian corpus luteum from degeneration. The corpus luteum responds by producing high levels of progesterone and estrogen, which maintain the thick endometrial lining and prevent menstruation. The baseline physiology of the body is now altered, and the gestational period has officially begun. Thank you for joining us on this academic review of human conception. Understanding these fundamental cellular interactions, cellular blocks, and endocrine feedback loops is vital for mastering embryology and clinical reproductive medicine. If you found this structured overview helpful for your studies or general knowledge, please hit the like button, subscribe to Repro Decoded for more detailed biological breakdowns, and leave a comment below with the medical concepts you would like us to decode next. We will see you in the next lecture.